Hello, I'm going to talk a little bit, kind of following on from the last talk about looking at relapse and reinfection and sequencing. But we've been developing a TB sequencing pipeline, specifically kind of for low middle income settings and resource poor laboratories, which have a slightly different way of doing things and way to build it up. So our question was, can we use whole genome sequencing to complement already routine diagnosis for TB in resource-constrained settings where costs are potentially more important than they are in other places, and especially for hard-to-treat cases, so in those relapse reinfection cases, and also sort of multi-drug resistance where people aren't responding to the treatments that they've been getting. So our aim was to build very much a pragmatic pipeline. So the idea was to balance cost, accuracy, obviously, because you need to know exactly what you're looking for, but also feasibility and sort of user friendliness of the pipeline. A lot of sequencing is bioinformatics, and unless you're sort of away with command lines, it can be quite complicated. So we were trying to make it so that if you went into a laboratory where you were doing clinical trials, any one of the people who were working in the lab with a little bit of sort of training would be able to go away and do this. So that was kind of the aim, quite a lot of aims, but to try and make it into one. So we chose to use Oxford Nanopore Technologies as our sequencing platform. I don't know if you've heard of it or seen them before, but there are lots of reasons why it's really useful. It's got a really cheap initial setup, so it's about $1,000 for the startup pack with flow cells and kits. It also has a very small laboratory footprint, and I have one here. Neil said it would be fun to do a show and tell, so I might send this round. It's also got a flow cell in it, so it's the device itself is the MinION, and then the little bit, the black one on top, is the actual flow cell where the sequencing takes place. So just pass it around. You can take this on my camera. So Nanopore sequencing is different to Illumina, which you probably may be more familiar with. So Illumina uses short read technology and Nanopore uses long read technology. And this is useful because it's much better able to span quite difficult, repeat regions instead of sort of having lots of little bits to kind of piece together. You have one long strand that covers the whole bit, or most of it, hopefully. It also has a relatively straightforward library preparation as well. And you can see these pictures that Nanopore have got. Move him again. So they've sequenced in space. They can sequence in the jungle in high sort of humidity. They've sequenced in Antarctica using a mug of coffee to warm up some of your reagents. They've sequenced in the desert. The idea very much for Nanopore is sequencing anywhere by anyone and anything. So hopefully this kind of fits our remit. But my screen's up. Screen's going the wrong way. There we go. So as I say, Nanopore uses long read technology. Illumina creates reads of about 200 to 600 bases. Nanopore technology creates bases of about somewhere between 1,000 and hundreds of thousands. So it creates much, much longer reads. And the way this works is the DNA moves through a pore on a charged membrane. So if you see on the Nanopore and it comes around, there's a little patch where it's got sort of liquid on it. That's where the membrane is, and it's got pores. The DNA moves through those pores. And as each base passes through that, it creates a unique change in the charge of each of the pores. And I think that might be better explained if I show you this here. So you've got the DNA here, it gets separated, and as it goes through the blue pore across the membrane, it changes the current. And so you get this kind of squiggle plot. There you go. And then the computer will change that into the A's, T's, G's, and C's, and that's called base calling, which is something that Illumina doesn't have to do, but this is a completely different way of sequencing. So we've come across a lot of problems, and if anybody has sequenced TB, you've probably come across them as well, and working with TB in general. So it's very slow growing. It's a category 3 organism, so you have to have specific lab equipment and levels. When you extract the DNA from it, because it's got such a complicated cell wall structure, it's very difficult to extract the DNA. I think a lot of sites that we've been working with use the CTAB method, and that often has a low yield. And with Nanopore, we found, because it uses generally native DNA, don't PCR it, you need quite a high level of DNA to begin with. And then the genome itself can be complicated because it has some very high GC content and, as I said before, many repeating regions. That's all well and good if you have a well-resourced laboratory, but when you then actually have to think about other things like power and electricity, the internet, this then starts to become a little bit more complicated again. So what we've tried to do is build up an entire pipeline from extraction to sequencing to sort of analysis all the way through. And at each stage, we've tried to optimize it. And as I don't have very long, I'm just going to concentrate on what we've been doing for the actual library preparation and comparing the different kits. So as I say, the TB extractions generally yield sort of low concentrations, although we've kind of, or Neil actually has been modifying it, and we've got much better concentrations now for sequencing. But because we started off with relatively low yields, and I thought it might be interesting to see what would happen if you did have low yields, we started off with a PCR-based kit. So the input was somewhere around 2 nanograms per microliter, so not very much at all. But what we found was the amplification of the whole genome wasn't very even at all. So this is RPOB, it's the rifampicin resistance. And you can see, so these black lines here are the reads, and they've all been sort of piled up on top of each other. And you can see that we've got a fair few in one part of RPOB and absolutely nothing at all in the other. And obviously, if you're looking for a SNP in a bit where there's no coverage, then you're not going to find what you're looking for. So that wasn't ideal. So that kit was discarded. We then went on to using the rapid barcoding kit. So this was native DNA. So we weren't PCRing it at all. We just needed to start with a lot more. So 400 nanograms, which worked out to be about 50 nanograms per microliter. And also, because it uses long read technology, Nanopore requests that you have around 30 kb, sort of the size of your DNA. So it's really important to use something like a tape station, bioanalyzer, or gel electrophoresis to make sure you've got that really good, nice DNA. Because if you have a lot of fragments, it kind of makes it a lot less efficient. However, having said that, and I can talk about this later if anybody wants to know, TB, I think, is forming primer dimers or sort of balls as it goes through because it's got so many repeat regions and it's kind of clogging up the pores. So we did an experiment and actually found that having somewhere between 10 and 20 kb was actually better. So don't tell Nanopore. We also found that if you normalized it for femtomoles rather than size and concentration, it worked a lot better because you can run multiple samples on each run. And so if you normalize it for femtomoles, because some might be 30 kb, some might be much bigger, you got much better number of reads for each sample. This gave us a lot better coverage. So you can see that there are reads across all of our RPOB, but it was still a little bit patchy. And what we've been doing recently, Alp, who some of you might know, who now works for Nanopore, recommended a new version of the kit called the Rapid Barcoding 96 kit. So as the name suggests, you can potentially run up to 96 samples, although probably not whole genome. You don't need as much DNA as an input. So he recommended up to 400 nanograms again, but you can put in as little as 50. And this is what I did for this first test that I did. And within 24 hours of sequencing, I had over 40 times depth for all of the isolates. So you can see that that's way better. And so the more depth you have, the more confident you are that each base is what you think it is. And so therefore it's much more accurate, which is something that has been leveled at Nanopore sort of compared to Illumina. So this kit is definitely much better. For the extraction, we decided in our sort of protocol to go with CTAB because that's generally what a lot of our sites tend to use already, although any extraction kit that you can get the right amount of DNA from is worth it. For the library prep, we decided for the native DNA, but the newer version of the kit, so you get enough reads per sample. With base calling, there's various different programs that you can use base calling to do base calling, but also different settings. So you have fast base calling, high accuracy, and super high accuracy. The trouble is the more high accuracy ones take up an awful lot of computer power. And if you don't have a very powerful computer, it takes days, like days and weeks. So you've got to, I guess, factor in how powerful your computer is, how long you want it to take until you get to your results. So high accuracy would be good, but if you don't have a computer that's kind of capable of that, then fast base calling with the newest version of Guppy, which is the base calling program, is going to work okay. We also then analysed different programs that you can use. So there are some command line ones and some cloud-based ones, and we found that TB Profiler seemed to work quite well. So that's the actual practical laboratory side of things. This bit is the things that I think maybe people don't necessarily think are going to be that important, but actually it turns out it really is. So product lead times and customs issues. I tried to take the MinION to a number of countries now and to do a number of different things with it. I was due to go to Zambia in October, and unfortunately, so everything arrived in Zambia on time. I had about 3 or 4 weeks. It was all there, it was all ready, but it got stuck in customs for 4 weeks, and we couldn't get it out. It was just, there was red tape. And so the MinION didn't make it to Zambia, and nor did I. But we're planning to go again in the new year and actually do some more training as well. So actually, it's probably worth it. Correct storage and reagents and flow cells also really important. And I know you're laughing because I know some of you know exactly what I mean by this. The kits are, you can store them in the freezer, although they have created a field kit which doesn't need any cold chain at all. The flow cells, and I cannot stress this enough, do not freeze them because they are a membrane. It's like a human, a human membrane, human pores, they just pop. And if you pop them, then you've wasted about £800 per flow cell, which is not ideal. So they can be kept at room temperature for up to a month, or you can keep them in the fridge for around 3 months. That's Nanopore's warranty, but actually they do tend to last a bit longer. But as over time, the number of active pores goes down, so the number of active pores corresponds to how efficient and how much data you'll get out of it. So the newer, the better, really. So please store everything at the right temperature. You need power for the duration of your sequencing run. If you have power cuts frequently, it might be worth getting both a UPS, so an uninterruptible power supply, and a generator if that's not already something you have in your department. You can run them off laptops if they're powerful enough, so you'd have that backup of the battery, but to be honest, sequencing on a more powerful computer is probably the better way of doing it. But you have to have power for at least, so I tend to run it for 48 hours, but you can run it up to 72 hours. So you need stable power for all of that time. You also need internet connection for all of that time, but especially at the beginning, because when you start a sequencing run, your computer connects to the Nanopore servers to download drivers and things like that. Unless you have the Mark 1C version of that Nanopore, which doesn't require the internet because it's kind of like a little handheld computer. You also have to consider the analysis programs that you're using as well. So if you're doing command line interface ones, then obviously that's just on your computer. But if you're uploading them to the cloud or to the browser, then you need the internet. And these files are really big. They're gigabytes worth of files. And so if you have a very slow internet connection, again, it's going to take you a long, long time. It did actually make it somewhere. We went to Tanzania and we ran a workshop, me and Sylvia, who is not here. We had about 20 people from all over East and South Africa from different sites, and they learned the whole of the sequencing pipeline, and everything was fine until we started to set up the sequencing run, and we didn't have good enough internet, and so there were people running around looking for Wi-Fi hotspots all over the lab, and it was chaos. And it took me 6 times to get it to start, and then it was fine. But a man had to stand there with his Wi-Fi hotspot for about 20 minutes to get it to work. So internet is also really important, but really, I think it's fine if it cuts out whilst it's running. It just really needs the internet when you first start. So yeah, internet's also important. And as I touched on earlier, they can generate an awful lot of data, so up to 50 gigabytes worth of data. You're getting sort of millions and millions of reads. So you also have to consider storage. So if you're making 50 gigabytes of data from one run, that's maybe 6 isolates. That's going to be an awful lot of terabytes after a while. So you'd have to consider maybe paying for cloud storage or a lot of hard drives. And if you run out of storage space on your computer when you're running it, it will halt the experiment as and when you run out. It's the same if the power interrupts as well. It will halt it when the power interrupts. So it's really important to have power, internet, and also enough space. Thank you to everyone who's been involved, Neil especially, because he's been very helpful, and all of the trials teams. Does anybody have any questions? Is that 10 minutes?
Linzy. it is dramatic. It is one of the two.
Oh yeah, I was just going to say, I was in a meeting with Nanopore the other day.
How are you?
Yeah, and they said exactly the same thing with you, that said that when they fragment their reads down, it will actually 10 to 20.
Is that Justin by any chance?
It was.
Yeah. I've been working very closely with that. It's been following me through this journey. And I went to a sequencing talk at the Crick, and there was a team there who had a very AT-rich protozoan plasmid, protozoan parasite genome, and he was having a very similar problem. And he said that it was probably because this thing had a lot of repeat regions, because what happens is when it goes through, this other sort of piece just sort of floats around. And I think what it probably does, a bit like with PCR, is it forms these balls and it probably gets stuck. It might be a combination. I don't think we really necessarily know yet, but yeah, certainly when I sheared them using Cavaris tubes, and somewhere between 10 and 20 actually, it's a trade-off between having obviously shorter reads and therefore less information, but actually it sequences better for longer. So TB is always a trade-off, isn't it? I think.